Characterization of biomolecules by electrophoresis. Electrophoresis is a technique used to separate and analyze different biomolecules based on their size, charge, and mobility in an electric field. This method is commonly used to study proteins, nucleic acids, and carbohydrates. In electrophoresis, biomolecules are placed in a gel matrix and subjected to an electric field. The molecules move through the gel at different rates depending on their size and charge. Smaller molecules move faster and travel further through the gel, while larger molecules move more slowly and travel a shorter distance. By comparing the migration of biomolecules in the gel to known standards, researchers can determine the size and charge of the molecules being studied. This allows for the identification and quantification of specific biomolecules in a sample. There are several different types of electrophoresis, each with its own specific applications and advantages. 1. Agarose gel electrophoresis. This type of electrophoresis is commonly used to separate and analyze nucleic acids, such as DNA and RNA. Agarose gel is a porous matrix that allows for the separation of nucleic acid molecules based on their size. Smaller molecules move more quickly through the gel, while larger molecules move more slowly. 2. Polyacrylamide gel electrophoresis. Page is commonly used to separate and analyze proteins. It can be used to determine the size, charge, and purity of protein samples. Page can also be used for DNA sequencing and the analysis of protein-protein interactions. 3. Capillary electrophoresis. This type of electrophoresis uses a narrow capillary tube filled with a buffer solution to separate biomolecules. Capillary electrophoresis is often used for the analysis of small molecules, such as amino acids, peptides, and small nucleic acids. It is a high-resolution technique that can separate molecules based on size, charge, and polarity. 4. Isoelectric focusing. IEF is a technique used to separate proteins based on their isoelectric point, pi, which is the pH at which a protein carries no net electrical charge. In IEF, proteins are separated in a pH gradient, and they migrate to their pi, where they become immobilized. This technique is often used for the purification and characterization of proteins. 5. Two-dimensional gel electrophoresis. 2D page combines two different separation techniques to achieve higher resolution and better separation of complex mixtures of biomolecules. It involves separating proteins based on their isoelectric point in the first dimension, followed by separation based on their size in the second dimension. Each type of electrophoresis has its own advantages and limitations, and the choice of technique depends on the specific biomolecules being studied and the research goals. Electrophoresis has a wide range of applications in various fields, including molecular biology, genetics, biochemistry, and clinical diagnostics. Some common applications of electrophoresis include 1. DNA and RNA analysis. Agarose gel electrophoresis is commonly used to separate and analyze DNA fragments, such as those generated by PCR or restriction enzyme digestion. It is also used for the analysis of RNA samples, such as mRNA or small RNAs. 2. Protein analysis. Polyacrylamide gel electrophoresis, PAGE, is widely used for the separation and analysis of proteins. It can be used to determine protein size, purity, and charge, as well as for the characterization of protein-protein interactions. 3. Clinical diagnostics. Electrophoresis is used in clinical laboratories for the analysis of serum proteins, such as albumin, globulins, and hemoglobin variants. It is also used for the detection of genetic mutations and polymorphisms. 4. Quality control in biopharmaceutical production. Electrophoresis is used for the quality control of biopharmaceutical products, such as monoclonal antibodies, recombinant proteins, and vaccines. It can be used to assess protein purity, identity, and post-translational modifications. 5. Proteomics research. Two-dimensional gel electrophoresis, 2D page, is commonly used in proteomics research for the separation and analysis of complex protein mixtures. It can be used to identify differentially expressed proteins in disease states or in response to drug treatments. Overall, electrophoresis is a versatile technique that plays a critical role in the analysis and characterization of biomolecules in various research and clinical settings. We hope you enjoyed learning about electrophoresis, their types, and applications. Don't forget to like, subscribe and share for more such fascinating scientific content. Thanks for watching.